I'm going to start off today with just a brief overview sort of the general protein expression field and highlight maybe some suggestions of what the particular organisms that we use are best for. And then I'm going to spend most of my time today talking about E. coli, and then the speakers after me are going to cover insect cells and yeast. So we're going to cover the whole realm of non-mammalian expression. But I'll touch on mammalian expression in a minute. So for those of us in the protein production field, the key features in how to make a protein, I think, are often summarized as Julius Caesar would have said about ancient Gaul, which is that it's really divided into three parts. There's expression of the protein, right? Can we make it? There's what we call solubility, which might be better in many cases be considered activity or proper folding, proper aggregation state. And then can we purify it to homogeneity? So I'm going to spend a little time today talking about all of these, though we're going to really focus a lot more on the expression and solubility components than the purification, although there's an entire track at this meeting that's focused more on purification issues. So some of the big issues for protein production, I'm sure most people here are familiar with, quality, and that covers a large range of things from what we call solubility, which often means what's the aggregation state of the protein, is it well behaved? How contaminated is it during the purification process? Is there proteolysis, either in vivo or in vitro? And are there post-translational modifications? And then the other big one, of course, that everyone's concerned about is quantity, right? What's the yield of protein? And that's not just how well can you express it, because we can express proteins often very well, but what is actually the amount of final purified product we can get out? And what is the amount of protein per volume of biomass? And then there are other issues like reproducibility and how well you can scale it up. And the bottom line for a lot of us is that if you have a lower yielding system, you're going to spend a lot more money trying to scale that up to make enough protein. If you have a lower quality system, then you're going to spend your money on purification optimization. And so we try to find systems which can balance these two things, get high yield and high quality. So there are four broad categories of hosts, and we're going to discuss, like I said, three of them today, all the ones except mammalian here. For anyone interested, there's a short course entirely on mammalian expression tomorrow afternoon, which will focus on transient expression in mammalian cells as well. So I'm just going to run really quickly through some lists of what I perceive as benefits and drawbacks of some of these systems, much of which will be expounded on by myself and other speakers later. So bacterial expression has a lot of benefits. It's really cheap. It's got a high biomass. It's very scalable, and it's probably the fastest growing organism of any of the expression hosts. It also does no post-translational modifications, and you'll see that shows up here in benefits and in drawbacks, and I'll talk more about this later. That can be a plus or a minus depending on what your situation is. The major drawback to bacterial expression is that bacteria are pretty poor at properly folding and producing heterologous proteins, so human proteins, mammalian proteins, and that leads to a major insolubility problem that I will spend a lot of my time talking about. Bacteria also produce endotoxin, which can be an issue depending on your downstream applications. So we use bacterial expression for obviously prokaryotic proteins, things that are small domains of proteins, anything that doesn't have a lot of complication like disulfide bonds, membrane proteins, glycosylated proteins, or if you don't actually care about the solubility or the folding. So coli is really great for making massive amounts of insoluble protein, which for many applications is quite useful. For instance, in generation of antibodies, that's really useful. Yeast are by far the most inexpensive eukaryotic organism to grow. They're very fast growers as well, and they produce incredibly high